Well, good morning and good afternoon, wherever you guys are logging in from. I'm Lisa Chen at UCSF at the Curry International TB Center, and I'm very pleased to welcome you to this webinar on advanced diagnostic topics. And I think we'll just hop right into it. We've got two wonderful speakers today in an hour long, and we'll just get going with the first speaker. So Dr. Aditya Kadamachi is a professor of medicine and epidemiology at the University of California, San Francisco. He's a great friend and colleague and has been doing research for the last 15 years on development, evaluation, and implementation of TB diagnostics in high-burden countries. So I think it's an excellent person to catch us up with what's going on in the field. And I'd like to hand it over to you, Aditya. Thank you.
Great. Thanks, Lisa. Sorry, let me just reshare my screen. Not sure what happened. Great. Can you all see the slides in the right view?
Looks good.
Great. Well, thanks everyone for joining. I'm excited today to just talk about advances in TB testing and next generation testing for pulmonary TB. This is a talk I developed and along with Claudia Denkinger, who's at the University Hospital Heidelberg and formerly was the head of TB at FIND. And so excited to share some of the slides with you today. So to start out, I don't have any relevant industry financial relationships to disclose. So in terms of learning objectives for today, so first I wanted to just go through the current recommendations and available tools for rapid diagnosis of TB in the United States and globally. And then talk about sort of improved knowledge of the progress and advances in development of novel diagnostics for TB, focusing on sort of molecular tests and sort of urine lab tests. I'm going to go through this quickly. I mean, I think this is not news to this audience that we have a large case detection gap of about 71%, which is now even thought to be larger in the most recent WHO report after the COVID pandemic. And novel diagnostics are clearly part of the solution to this problem to improve the speed of diagnosis, the accuracy of diagnosis, and particularly in high burden countries, making diagnosis available at lower levels of the health system. This slide summarizes current guidelines for TB diagnosis, both from the ATS, Infectious Disease Society of America, and CDC, which were last updated in 2017, and then the WHO consolidated guidelines, which were last updated in 2021. So in terms of the US guidelines, sputum smear microscopy, there's still a strong recommendation for that, as well as for liquid and solid culture. Molecular testing for TB diagnosis has a conditional recommendation, but a strong recommendation for molecular testing for rifampin, with or without INH resistance. So when we look at the WHO guidelines, I mean, I would say they've in some ways advanced, beyond what we're doing or recommending in the US, right? So rapid molecular tests are now recommended as the first line test as a replacement for smear microscopy. So really smear microscopy is no longer in the international guidelines. Universal testing is recommended for rifampin and INH with or without INH resistance. And then, we have urine lab testing as an option, in particular for HIV positive inpatients, and as a conditional recommendation for outpatients. In terms of how we can implement these guidelines, this slide summarizes the current options for actual sort of testing platforms that are available. So in the United States, the expert MTB-RIF assay is the only kind of rapid or semi-rapid or semi-automated assay that's FDA approved. And there's also sort of two more moderate complexity sort of laboratory assays that are molecular assays that are approved in the US. Again, globally, we've started to go sort of beyond this, right? So in addition to expert MTB-RIF, I didn't put on the slide, but there's the MTB-RIF Ultra cartridge, which is enhances sort of diagnosis, both sensitivity as well as specificity for rifampin resistance relative to the original cartridge. TB LAMP is a loop-mediated isothermal amplification platform that from Aiken, a company in Japan that has a conditional recommendation. And then most recently, I highlighted in red, there's another company from India called MolBio that had their assay TrueNat MTB, MTB Plus, and MTB-RIF endorsed by the WHO, and then as well as sort of 4 moderate complexity assays. So a quick word on some of the newer assays that people may or may not be familiar with. So TrueNat, again, is from a company from India. It's an automated battery-operated device for DNA extraction and PCR. The PCR itself is done on a disposable chip. So, for the sputum sample, similar to Expert, you add a reagent, put it into the extraction device. and then load the extracted material onto the cartridge, insert the cartridge into the reader, and it produces results in less than 1 hour. So this is similar to Expert, and in some ways even more so than Expert. This enables molecular diagnosis to be done even at lower levels of the health system in high-burden countries. In terms of performance or accuracy, there was a large trial, including 1,800 participants in 19 sites across 5 countries. The data are shown in the figures on the right, but what you'll see overall is that the performance is very similar to Expert. So this is really the first serious competitor to Expert MTB-RIF. It's obviously being scaled up in India and now also in several other high-burden countries. In terms of moderate complexity tests, I'm not going to go through all the data, but there's 4 of them that were endorsed in the most recent WHO guidelines. And what I really want to highlight with this slide for the US context is, can we leverage economies of scale, right? We all know that molecular testing capacity has been increased, many-fold as a result of the COVID pandemic in the US, also sort of globally. And can we leverage that testing capacity to also sort of test for other diseases like TB and enhance molecular testing and rapid diagnosis? And how can we do that? So I think that's an important discussion that we need to have in the US as well, as we kind of aim to improve diagnosis over sort of microscopy. So in terms of looking forward, as I mentioned, what we have now in 2022 is, more options for moderate complexity PCR, as well as, low complexity testing with the TrueNat assay. I think moving forward, over the next few years, what we're going to see is investigation into alternative sample types, more point of care platforms, interest in cell-free DNA, which I'll highlight in a bit. And then, moving all the way toward, really kind of point of care sort of biosensors to detect molecular signals of TB, including cell-free DNA. So in terms of novel sample types, there's a lot of interest in moving beyond sputum. Sputum is difficult to collect. It's often not able to be collected from children and other populations without sort of more invasive sampling available. And so there's a lot of interest in sort of, can we diagnose TB using more easily collected samples like breath or oral swabs, blood, urine, and in children, perhaps stool. So I just wanted to go over, in particular, sort of some of the advances in stool, swabs, and urine based testing, and others we can get into if there are questions. So many of you may know, stool PCR testing has already been endorsed as a novel sample type for children by the WHO. The problem is sort of extensive. TB is extremely common in children, with about 1.1 million children developing TB and having a high mortality. And there's really no effective test that can be performed on easily accessible samples, right? And things like gastric aspiration, nasopharyngeal aspirates are not always sort of possible in high-burden countries. So several groups have worked on developing stool processing kits for automated extraction of DNA in stool for expert ultra testing. And what they show is, moderately good performance, right? Sensitivity around 50% with sort of high specificity, which is a large improvement over sort of, microscopy, both in terms of sensitivity, but also yield when you consider that stool can be much more easily collected from many children than sputum. And so this was based on these data. This is sort of endorsed, and now there are groups trying to sort of further improve sort of performance of sort of stool-based molecular testing. There's been a lot of interest in oral swabs. I think obviously the whole world is used to sort of swabs now with the COVID pandemic, but it's non-invasive and rapid sampling, and it's simpler processing relative to sputum. For TB in particular, Jerry Cangelosi's group at University of Washington has really showed that tongue swabs are much more sensitive for detecting TB than other types of swabs like nasopharyngeal swabs or buccal swabs. And so the data on the slide are from work that we've done in Uganda, where we collected two tongue swabs from participants, put them in a TE buffer, and then put that buffer into the expert cartridge. And what we're showing with that method is that we achieve pretty good performance, right? So overall sensitivity around 77% relative to sputum expert ultra, around 72% relative to culture with high specificity. if we look at the results a little closer, you see that, while the overall top line sensitivity looks pretty good, the bacillary burden is always lower with tongue swabs relative to sputum. So when the sputum samples are trace or very low, in terms of having a lower bacillary load, they're not detected by the tongue swab method. And even when the sputum expert ultra results are medium or high, the corresponding result for a tongue swab is lower, right? It's either low, very low, or trace. So what this suggests is that, again, with this current method of 2 tongue swabs and expert testing, we do well, and it could be a replacement for sputum testing when sputum is not available. But there's room to improve the assay for possibacillary specimens in particular. So one of the things that we've been doing in Uganda as well is to say, well, is there really TB DNA on the tongues of most people with TB? And so we've been working with Global Health Labs and our team at Walimu in Uganda to sort of address this question by really developing kind of a reference or lab-based assay, not worrying about a point of care format, and evaluating different sort of sampling devices or different types of swabs, different sample collection methods, different lysis and extraction methods, and different sort of ways to sort of amplify and detect DNA. And what we did is put together kind of the components that we thought were best or performed the best into a kind of a reference assay. And the results are shown on the bottom right. And what you see, again, the numbers are small and we're still enrolling more participants, but we're actually getting much better performance than with the oral swab expert ultra results I showed you earlier. Really getting at this point 100% of sputum expert ultra positive samples and maintaining sort of very high specificity. So I think these results are early, but they do suggest that TB DNA is indeed present, and if we can optimize sort of swab-based testing, it could be a potential sort of alternative to sputum moving forward. Just really quickly, I just wanted to highlight, other ways of detecting sort of TB DNA. There's been a lot of interest in breath aerosol, both because it's easy to collect by a non-invasive sampling like a face mask. And there's also this thought that there's a link to infectiousness, right? So those who have either live bacteria or DNA in breath aerosol are thought to be sort of more infectious, perhaps. It is just, I'm not going to go through the whole study, but very early, a small pilot study of breath aerosol sampling in the context of community-based active case finding in South Africa. And I think the interesting thing that they found was that there were a number of participants who had positive breath aerosol results, but negative sputum results at baseline. And when they followed up these participants at 6 weeks and 20 weeks, nearly all of them eventually developed a positive sputum result or other evidence of TB, like a positive PET CT, and all were sort of ultimately started on treatment. So again, just some very early data to suggest that breath aerosol may pick up a different type of patient and might pick up patients earlier than even sputum-based testing. But obviously, these results need to be confirmed in larger studies. The story is similar for cell-free DNA. It has a lot of appeal. I think many of us know it's transformed the diagnosis of cancer, prenatal diagnosis, and a few other areas. And again, it's appealing because the idea here is that it's not limited to disease that's in the lungs, right? So DNA fragments can be released from TB bacteria that are at any site of disease in the body, whether it's in the brain or lungs, kidneys, liver, etc. Right, and when those fragments are released into the blood or urine, the idea is to be able to detect them with a PCR-based assay. So there's been a number of sort of early studies looking at this, and as you can see on this in the graph, the studies really right now they're showing variable performance, and so there's a lot of work that needs to be done to sort of optimize these assays. I do want to mention that there is one CLIA certified cell-free DNA assay by a company, Carius, and it's a single blood test that detects cell-free DNA from over 1,000 microbial pathogens, including Mycobacterium tuberculosis. But overall, I would say it has limited use as a routine TB diagnostic, right? As shown in the data on the slide, it only has moderate sensitivity in smear-positive TB, 40 to 50%. And poor sensitivity in smear-negative TB, right? So it didn't detect any of the smear-negative cases. So again, this is not ready for prime time, but a lot of work is being done to sort of optimize the pre-analytical steps to see if we can improve performance. So what about LAM, right? And the big question for urine LAM has been, what about people without HIV? And so this was a nice study done by Broger and colleagues in 2021, where they showed that the current sort of LAM assay, that written here as LF-LAM, which is the determined lab assay, performs very poorly, as we know, in people living without HIV. So they looked at two new assays, the Sylvamp LAM assay, which was a second generation LAM assay developed by Fujifilm, and then kind of a laboratory-based, ultra-sensitive ELISA-based LAM assay to say, can we improve performance even better? And what they really showed was that both with the second generation point of care assay and the lab-based assay, both sensitivity and specificity actually improved, right? And so these were kind of intriguing data to suggest that, LAM is present even in many people who do not have HIV, but present at low concentrations. So a lot of work is going now into, can we develop third generation assays that can lower the limit of detection, right? And so the Gates Foundation has really been leading this work to couple better knowledge about LAM structure and epitope through epitope mapping with better antibodies and higher affinity antibodies, and then better abilities to capture, concentrate, and detect LAM in urine samples. And so there's a number of companies who are expected to come out with sort of third generation assays, putting together this kind of a best of the best approach that's been worked on over the last several years. And so we're intrigued to see, whether or not, performance can be further improved. So last, I'll just highlight that, in addition to kind of these TB specific developments, I think, one of the exciting things in the space of diagnostics is just the tremendous innovation that's happened to enable point of care testing, largely as a result of COVID investments in COVID diagnostics. And these are sort of really improvement in LAMP platforms. Many of you may know that there are even LAMP-based PCR platforms for home testing at this point. There are now even instrument-free molecular diagnostics for COVID, and then instrument-based immunoassays that really sort of improve analytical sensitivity. So there's a lot of interest in sort of getting some of these companies to pivot to TB as, the COVID-19 testing market becomes saturated. So that's sort of a focus for the diagnostics field moving forward. So I'll end by just summarizing that, at this point, I really want to emphasize that molecular testing should be the current standard for diagnosis of TB. There's a number of available options, particularly internationally, but still for low complexity tests, just the expert MTB assay available in the US. Innovations in sample type and platforms are anticipated over the next few years. I think you're going to hear a lot more about tongue swabs and mass-based aerosol, as well as cell-free DNA. And as I mentioned, we're excited to be able to try to leverage the point of care platforms that have been developed for COVID, and to sort of add TB testing capacity to some of those. And last, urine LAM testing is showing increasing promise, and there are third generation assays that are expected to undergo trialing in 2023. So we're eagerly sort of waiting for those results as well. So thanks for listening, and happy to take questions.
Great. Thank you so much, Aditya. I think we've got some questions for you. I think, I heard this talk at ATS this year, and I was so excited just to hear everything like rolled up into one quick piece. It's really food for thought. And Susanna Graves from San Francisco is asking regarding breath aerosol. For the patient at the top of the chart who had alternate diagnosis, was there environmental contamination or perhaps any picking up from another patient or contact? I'm not too sure what you mean by the top of the chart, Susanna, but I'm seeing you're adding to this, wondering if there would be application for pinpointing exposure in an ongoing transmission situation. So is there a chance of environmental contamination when getting a breath aerosol? Well, I guess maybe it's better to explain how. I've seen some of the explanation about how breath aerosol is captured, but I'll let you go ahead and explain it.
Yeah, sure. I think, with any TB testing, contamination is always a sort of concern. I think with breath aerosol, the way that it's being captured is actually sort of a face mask, put over, a person's face, and there's a disc at the end of it which sort of captures sort of aerosol. And so, is it possible to sort of entrain some room air, while that's happening? I mean, I think, it's certainly sort of a possibility, but I think it's sort of no more likely than if you collect a swab or even a sputum sample and, you're able to sort of transfer, a DNA is sort of transferred. I think more of the contamination is likely to happen because, for any molecular test, because there's DNA present in that laboratory environment. So the closed systems for testing are really important. And that's sort of, what the GeneXpert platform does, for example. It's a closed cartridge system. And so I would be more concerned about sort of open systems. That's been one of the concerns about sort of the TrueNat sort of platform, that it's not as closed a cartridge system as the way that the GeneXpert is. And so for whatever a molecular sort of sample that's collected, I guess I'm more worried about the actual testing process than the sample collection for most of them.
There's a couple other questions we could probably hit quickly. I just for clarification, someone just needs to know what is LAM exactly?
So LAM is a lipoarabinomannan. It's a antigen in the cell wall of Mycobacterium tuberculosis that's specific for MTB, and so it's a kind of a lipoprotein that gets shed from the cell wall of Mycobacterium tuberculosis, and is therefore and then can be detected as a protein in clinical samples.
All right. Hope that answers your question. And then Nino was asking, is saliva good enough sample for smear microscopy or expert? The fact it's come from the mouth too and easy to get collected.
Yeah, I mean, I think it's an interesting question, right? I mean, I don't think it's going to have the same yield as either sputum or tongue swabs. The TB, I mean, the mechanism of how sort of TB gets deposited on the tongue, I mean, it's not that there's TB in the oral cavity. It's that when you cough and patients cough, that the back of the tongue acts like a kind of a barrier where a lot of things don't always make it out, into the anterior part of the oral cavity. And so you have, things that are deposited there when people cough. With saliva itself, yes, I mean, we've definitely seen TB DNA in saliva, but I mean, it's, I think without sort of actually scraping the tongue, you're unlikely to get nearly as much DNA because it's otherwise purely a upper respiratory sampling type. And unlike viruses, TB is not really thought to be present in the upper respiratory tract.
All right, hope that helps too. I think that makes sense to me. There's going to be a lot more questions here than we can get to, but maybe Diffie, when you're done, you can see in chat or in the QA some of them. But I definitely want to get to this one in the QA, which curious if you could speak a little more to the Carious test, because we've seen it used more in practice recently. What do you think about it in terms of reliability? And I know this is true, particularly in Southern California. I don't know where folks are asking this question from, but it does speak to a lot of the crowd here. I think more and more academic centers, ID specialists are sending Carious for other reasons, and TB is coming up incidentally. But what do you think about the future for Carious as a reliable test for TB?
Yeah, I mean, I think I don't know enough about the and that they've done enough work to show the specificity of the assay for TB. The data are still sort of small, and so how reliable it is when you have a positive result, it's hard for me to sort of speak to. And I think you would have to I think I see this as more of one of those tests where you have a fever of unknown origin, or you don't know what's going on with the patient, and you just like you send off a universal PCR, you can send off a Carious test to kind of just get a sense of what hits come up, and then you have to use that in along with your clinical context to make decisions about treating the patient or not for TB. And so I think that's going to depend on what your pretest probability is, what other diagnoses are on your differential, and what the likelihood of them is. It's certainly not reliable enough when it's negative to say that it doesn't have TB based on the sensitivity data that I showed you. And so, it's hard for me to answer it more definitively than that. That I think it's another piece of information that you get clinically that you have to decide what to do with, and that decision may be different depending on your estimate of the pretest probability of TB in that patient.
Yeah, I think it really is interesting in that it has been coming up as kind of a incidental finding when there's a large differential diagnosis. So I guess that's, I guess we can see that as the utility, but it certainly isn't the test you're going to reach for first if you're considering TB as your primary suspect. Is that about what you're thinking, Aditya? Yeah, there's so many here. How about this one? I love the work on additional sample sites like oral swabs that can be used on an expert that we have in the US. Because so many recent tests are not planning to go through FDA approval, is there a way to move advocacy or other work to try to get more of these newer technologies approved in the US? I worry that like Ultra and Lamb, we might never get access to cell-free DNA or aerosol technologies if they're ultimately successful for TB diagnosis. Any thoughts on that?
I worry about it too. I'm not most of my work is focused in high burden countries. I'm not a laboratorian in the US, and I don't know if there's any laboratorians sort of on the call, but what it takes to get something to be CLIA waived or to be done with kind of, there are a number of tests that are done in the US that are not FDA approved, but are, laboratory validated, tests. And so is there a pathway to taking some of these tests that might be, endorsed by the WHO and then having individual laboratories validate them in their own laboratory context, and what that pathway looks like? I'm not sure, but I do agree that it's going to be a challenge moving forward as the testing options continue to sort of expand internationally. We're going to get more and more behind in the US.
Well, I think the good news is our next speaker is a laboratorian. We could get pick her brain, but also I think it really does take a lot of advocacy from our own public health groups. A lot of the companies aren't necessarily applying for release in the US, and I think that that's where we need to show that there's actually demand. Is the rough part. But so what a great segue to our next speaker. And thank you so much, Aditya. I know how busy you are. We're so glad we caught you between plane flights. And so I'll go ahead and move to our next speaker, Dr. Agnieszka Martynova-Pekova. She's the lead of the TB Reference Laboratory at CDC and oversees the Molecular Detection of Drug Resistance Service, or a lot of us call it the MDDR. We're so thankful for that, Agnieszka. She joined the CDC Division of TB Elimination in 2019 as a senior microbiologist and has been in her current role since July of 2021. Agnieszka also serves as the CLIA technical supervisor for all test systems performed by our CDC TB Reference Laboratory. And we're very pleased to have you join us and really tell us what's up next coming down the pike. I think we're all excited from your CDC lab. And then maybe at the end in QA, you could talk about these other tests or your take as a laboratorian on whether or not we'll ever get access in the US. But you're welcome to share your slides, Agnieszka. Thank you.
Thank you. Do you hear me and do you see my slides? I think I tried
to share my
screen.
You, but you'll need to try again with your screen share. Okay. Terrific. You're all set.
All right. Well, good afternoon, or maybe I should say good morning to those of you that are on the West Coast. And thank you, Dr. Chen, for inviting me today to talk a little bit about the transition of the molecular detection of drug resistance service towards the at CDC towards the use of next generation sequencing. So the MDDR clinical service was first implemented by the CDC TB lab back in 2009. It offers rapid detection of drug-resistant TB by DNA sequencing. We have two methods that we use for DNA sequencing, and we're currently using them. It's the screening pyrosequencing for rifampin and INH resistance, and then we have Sanger sequencing for first and second line drugs. Also, for any clinical sample or isolate that we receive for MDDR, we attempt phenotypic drug susceptibility testing in parallel to the via molecular testing. This service is available to all 50 states, U.S. territories, and U.S. affiliated Pacific Islands. It's free of charge, and the shipping cost is covered by the FedEx account that is managed by APHL. We also offer, as part of this MDDR service, clinical consultations whenever they are requested by our submitters for interpretation of the laboratory results that we release. The acceptable sample types for MDDR are confirmed pure MTBC isolates, but also mixed and non-viable cultures that are confirmed to contain Mycobacterium tuberculosis complex, as well as MTBC not positive processed sediments. Although we do not directly accept tissue samples to work with, we partner and collaborate with the CDC's infectious diseases pathology branch. And our submitters, if they have fixed tissues that meet the criteria, the acceptance criteria of IDPB, they can submit their samples there. And if MTBC is confirmed, IDPB can forward to us DNA extracts, and we perform pyrosequencing for rifampin and INH. In 2021, we have tested a total of 912 samples, and 21% of those were sediments that were submitted to us. The mean turnaround time for the service for last year was 4 days. So why do we need to transition towards next generation sequencing? First, I just want to clarify the terminology in case there's someone in the audience that's not familiar with this. When we say next generation sequencing, this is a term used for the platform or the methodology. The targeted next generation sequencing that I'm going to be talking about, as well as whole genome sequencing, these are assays that utilize the next gen sequencing platform. We often get the question, why don't we want to transition towards whole genome sequencing? But at this point, we are focusing on targeted next generation sequencing because we want to be able to test both sediments and isolates. Whole genome sequencing is in our future, but not immediate future plans. Some of the reasons for us to need this transition is that we would like to increase the output per run. We would like to expand some of the genetic loci that we have been testing, but also to add loci for new TB drugs. We want to be able to identify novel mutations and to better detect heteroresistance, specifically in sediments. And the bioinformatics pipeline that is going to be used for the TNGS data analysis, it's going to limit to a certain extent the human error in the analysis portion. We're also in certain ways forced to think about new methods just because the pyrosequencers that we are using, the 96-well platforms, are going to be discontinued by QIAGEN at the end of 2023 when this method is apparently going away for us. The targeted next generation sequencing that's going to be used for MDDR service, it's going to be a deep sequencing of PCR amplified drug resistance loci, and we're going to be using the Illumina MiniSeq platform for this assay. It is suitable for both sediments, as I already mentioned, and isolates. With our current template, we can accommodate up to 10 clinical patient samples on a run, plus 2 controls that we are adding to each run, so a total of 12 samples. And we are only sequencing about 0.3% of the genome of MTBC, so pretty small portion. But with this assay, we are going to be looking at loci that are contained in 16 genes, and we're going to be offering information about at least 12 TB drugs. The targeted next generation sequencing, just very briefly, has 2 parts, a wet lab portion and a dry lab portion. And the wet lab begins with the heat killing activation of the sample that we receive. So that way we prepare a crude DNA prep that we then use as a template in our PCR reactions. We perform 24 PCR reactions for each sample, or for the total of 12 samples, we are going to have in each run 288 PCR reactions. We then pull all the amplicons for each particular sample. It undergoes several steps as purification, normalization, etc. Then all the individual 12 libraries are pulled together and they're loaded into a cartridge, and then this cartridge is loaded into the miniseq instrument, which runs for about 20 hours. And the reason why I'm listing this is just to tell you that this process takes about 2 days to be completed. And then we also, after this is completed, we come to the dry lab portion of the targeted next generation sequencing. So what the output of the Illumina sequencing machine is FASTQ files. We check the quality we check the quality metrics for the run, and if everything is okay there, then we feed those FASTQ files into the custom-made bioinformatics pipeline that is going to analyze this data. It undergoes through several steps, including decontamination, trimming, and then it aligns it to our reference sequence, which is H37RV sequence. And then it calls the variants, which are the mutations found in the regions of interest. It also gives us information about the data coverage. So basically, we have established a number of metrics at the run level, sample level, as well as the mutation level. And we just want to make sure by doing this that what you get as a submitter is accurate information and as well as is of a highest quality. I'm showing on this slide the Sanger sequencing current panel in the left table compared to the what TNGS panel will look like. So we're going to have information about the same drug groups as Sanger, including rifampin, INH, ethambutol, pyrazinamide, fluoroquinolones, and second-line injectable drugs, with the only difference that the TIL-A locus, which is specifically for capromycin, is not going to be included in the TNGS panel. But instead of that, we are getting now information for drugs like linezolid, bedaquiline, clofazimine, and specifically the CAT-G locus, which we are currently only testing the region that includes the 315 codon mutation. Is now going to include the full-length gene for CAT-G. This is the rubric of our reporting rules. So basically, the type of data that we are reporting is going to be similar to what you are used to see coming from us, with the addition of this, the information for these new drugs. There will be some differences though. For example, for rifampin, we're now going to be only reporting mutations found in the RRDR 81 base pair region that is also covered by the gene expert, and codons 170 and 491. But any mutations outside of these reportable regions are not going to be reported. We're also including a minimum reportable alternate allele frequency threshold for the analytic pipeline, which is going to be 10%. So any variants that we see below 10%, even if they are otherwise reportable, are not going to be reported. These are just quality metrics that we have implemented. The MDDR algorithm that you're used to is not going to change significantly. One of the changes that you will notice is the turnaround time with the TNGS, and the time that is necessary for processing, for analysis, and all of this for a certain run is going to increase to approximately mean 7 days. But it's kind of a trade-off because now you're going to get additional information for other drugs as well. And another thing that we're currently working on is that we are working with the CDC ELIMS group to build out reporting of the MDDR results through the ELIMS system. So eventually we will discontinue faxing our reports, and those will go electronically through ELIMS, but this is really a very labor intense and kind of a cumbersome process building this out, because we are the pilot lab at CDC who is building this process in ELIMS for next generation sequencing, kind of a DST assay that includes number of drugs, number of loci, and a large number of reportable mutations for each of those loci. This is just a screenshot if there are laboratorians on the call that are interested in validation implementation of next generation sequencing assays in their laboratories. This is the CDC quality initiative, and it has great resources that are available for people outside of CDC as well. So to us, when we perform this TNGS validation for MDDR, we had the following objectives, and we wanted to answer a few questions: whether this new assay is going to be specific for MTBC, whether it generates results that are comparable to what we already have via Sanger sequencing, whether it can detect all type of mutations, not just substitutions, but also insertions and deletions in the genome, whether the results are reproducible, meaning different analysts can produce the same results, or on repeat we continue to get the similar results, and then whether it can detect mutations in new loci that we did not have existing information for previously. And one of the major kind of the things we needed to make sure during this validation process was the TNGS specificity. We know that in the samples that we receive, in probably 99% of the times, the contaminant, if it's a mixed culture, which we accept for MDDR, or if it's sediment, in 99% of the times the contaminant would be a non-tuberculous mycobacterium. So what we did in attempt to prove that this is highly specific, we took 9 pure cultures of NTMs along with a BCG control and H37RV MTBC control, and we ran those as part of our TNGS panel. If you look at the bottom, if we run all of this with a 16S RNA PCR assay, they all get positive. But what happens with the TNGS is the question. So we know for sure that we have some of the amplicons, the majority of amplicons, that are highly specific for MTBC. For example, the CAT-G amplicons, the fluoroquinolone associated JAR-HRB amplicons, those are highly specific. They do not basically amplify non-tuberculous mycobacteria. However, there are some highly conserved regions in the ribosomal RNA coding genes like RRS, RRL, as well as the RPOB gene amongst the mycobacterial species, and therefore it is possible that the PCR primers that we are using, as we are showing on the top kind of a graphic, it is possible that the PCR primers can amplify NTMs. But we do take care of this in several different ways. So first of all, NTM sequence in the loci and regions of interest, for example, in the RPOB, RRDR region, they have significant differences. They have large number of mutations if it's NTM compared to the Mycobacterium tuberculosis complex, and we are able to see those. The other thing is that running this data through the pipeline has a decontamination step, so it compares the data to a known database of non-tuberculous mycobacteria, and it gets rid of them early in the analysis process. And also, one thing I should point, with this experiment we were using pure NTM cultures, versus for MDDR we do not accept pure NTM cultures. At the worst case scenario is that this is going to be a contaminant that is coming along with the MTBC. So what we report, we know at the end of the process, we know it's MTBC. During our TNGS validation, we compared the results that we had with this existing Sanger sequencing data. We used a total of 106 samples that were originally submitted for MDDR, and we did have results for those, plus 11 WHO isolates for which we had whole genome sequencing results. And we made sure that 21% of this were sediments, just to resemble what we see in real life in the lab. And for the reportable mutations within the directly comparable regions, which were 12 loci between Sanger and TNGS, the results matched at greater than 99%. So we know that the TNGS is at least as good as the Sanger sequencing, and it's actually supposed to be better because now we're going to add even more information to it. We were also recently updated the performance characteristics of the MDDR service. For this purpose, we used about 5,000 information for 5,000 samples that we have run Sanger sequencing on between 2012 and 2021. And we compared those molecular results with the phenotypic results that we have here from our lab. So the updated kind of a performance characteristics, specificity and sensitivity are shown in the table here. And only a couple of things that I would like to mention. For rifampin, this specificity of 91.8% is likely due to those isolates that have low-level rifampin resistance, or those that are also known as disputed mutations, because those isolates quite often test as susceptible by phenotypic DST methods. In terms of pyrazinamide, you will notice that the sensitivity for the PZA is pretty low. It's lower than 70%, and this is likely impacted by the clade 1 isolates that we have tested during those years. This is the Indo-Oceanic clade. Those isolates usually test as PZA resistance phenotypically. However, they do not possess PINK-A mutations. And this, combined with the poor reproducibility of the midget PZA test, contributes to this low sensitivity. So based on these results, some of the interpretation comments that we'll be using for the TNGS moving forward are going to be updated as well. And we will likely regularly update our language that we use for interpretation based on the new data that we will accumulate when we start using the TNGS. In summary, the targeted next generation sequencing that we are going to implement is reproducible and robust. It will successfully replace the traditional sequencing methods for the MDDR service in early 2023. It makes possible detection of heteroresistance early, specifically in sediments, and it may be customized to include additional loci in the future. The reports will also be issued through the ELIMS system. And I know that this is a burning question. We get a lot of emails about this. We fully understand the necessity of having broad microdilution MIC method that is validated and be able to test patient sample with. This will likely happen after we go live with the TNGS. Once this is approved under CLIA, we'll be able to test patient samples and to report results for rifampin, bedaquiline, linezolid, clofazimine, as well as the fluoroquinolones, levofloxacin, and moxifloxacin. And with that, I just would like to acknowledge all the people that I have listed on my slides, specifically the applied research team within our branch that helped us tremendously during this process of implementation. Basically, they did develop the TNGS assay. They developed the bioinformatics pipeline that was custom made and we're going to use, as well as people on my team who participated in the validation of this assay, former employees here at the branch who have contributed towards the final results, and of course the office of my branch chief and system groups, Angela, Tracy, Lois, and Juan, without whom I wouldn't have made it. Thank you so much for your attention, and I'm happy to take questions.
Ataneska, thank you so much. You know, we're all waiting breathlessly for this new stuff to start. And I think we're particularly interested in having rapid access to bedaquiline and linezolid testing. So we're very anxious, and I appreciate the extra mention about being able to get MICs back online through your lab. I'm sure folks I'm seeing a big yes in the chat. We're all waiting.
It will happen. Believe me, it's a kind of a tough process to go through and get approval under CLIA, but unless we get that, unfortunately, we're not going to be able to test. So we have wrapped up the validation process, but it takes a lot of extra steps that needs to be taken before we can present this to the CLIA director for approval. But we're working on it.
Understood. And I think John was just asking just for, again, the timeline, timeline for when you think the next gen sequencing is going to be up and running, and timeline when you think the MICs will be up and running.
So with the TNGS, the packet have been submitted to the CLIA director. We've received and addressed feedback that we've received so far, and the approval is pending. We are hoping to get this, to go live with this very early 2023. We're hoping for January, but I cannot promise anything until we see the final approval from the CLIA director. We're just hoping for very early 2023. And as we go live with this, we are in the process of preparation of the MIC packet to also submit that to the CLIA director. So a little bit later in 2023, but we're hoping that both will be up and running in 2023.
All right, terrific. And I see that it looks like there are a couple of people with their hands up. Marlena, your hand might have been up prior for Aditya, but if you want to go ahead and unmute yourself. Marlena, would you like to answer a question? Maybe not. How about Lena Dominique? You also have your hand up. You feel free to go ahead and unmute yourself, and we'll go ahead and ask your question. It's not unmuting. I think
she should be able to talk. So
this might not be. Lena, are you trying to unmute? Well, while you're trying to figure that out, there's some other questions coming up. When will be adding pertometed? When will a pertometed loci be added? Now there's a question.
I cannot answer this question at this point. We are we are participating in a predominant study, but again, this would need to go, we're planning for it, but I just cannot give a timeline for this one. We're planning for it.
And even your sense of reliability for the mutations for bedaquiline and linezolid, what's your take on those? What's your take on the reliability of the loci that we now have identified for bedaquiline as well as linezolid?
We have very little data. I am not comfortable with saying, any reliability at this point. That's why we need to have both eventually to accumulate data using the next generation sequencing. And then once the MIC becomes available and we start testing those MDR isolates in parallel, we will accumulate inevitably more information. And with time, we'll become more confident in what may mean phenotypic resistance and which mutation would not. But what you should expect from us at the beginning, these are all going to be novel mutations for us. We just don't simply, first of all, there are not many resistant isolates available. So that's why even validation of assay like this is very challenging, because you don't have anything to compare to. But also initially we will be reporting as effect unknown until we have sufficient data to comfortably and confidently say that, yes, this means perhaps resistance.
Well, I've got a couple of extra, thank you. I've got a couple extra questions here from Demetrius. So number one, for pan-susceptible sediments and isolates, do you still perform phenotypic DST?
We do. We will
Yes,
continue to perform our agar proportion. We do this for every single clinical specimen and isolate that we receive, even for those that are submitted as mixed cultures. We do perform phenotypic in parallel. We do agar proportion testing for first and second line drugs and MGIT PZA. As well. We'll continue to do this, and in the very near future, we're planning on updating our agar proportion panel as well a little bit. But in terms of MIC, it's not going to be performed on every single, like specifically on pan susceptible, unless there is a specific request and need for that.
And the same participant just asked, and I think, of course, I think I know the answer is there going to be an associated cost with targeted next gen or MIC?
No.
No, CDC,
we're a government.
public health. So that's
Yeah.
Wonderful. How about Nino is asking, bedaquiline and linezolid, mentioned, I guess we said, and we did answer this at the beginning, that there's, will pertometed be in the current testing too? And it's just not
Initially.
Not yet. All right. In the we've got in the chat, Aditya, you're still on. Lisa Pascapella is asking, Ed Desmond always emphasized that smear status was an indicator of infectiousness. Will any of the newer PCR-based or non-PCR diagnostics also have a quantitative readout that indicates the level of infectiousness of the host?
Yeah, thanks. I mean, I think, Lisa, the smear status largely correlates with infectiousness based on bacillary burden, right? And most of the molecular tests, including the expert assay, have a similar sort of semi-quantitative readout. And the presumption is that, the higher the bacillary load, the more infectious they're likely to be. But of course, there's also lots of data, and we know that smear is not, and bacillary load are not sort of perfect correlates of infectiousness, and that there are many smear negative patients who transmit. And so I think overall we just need better tests to identify which patients are the most likely to transmit. But I think that the short answer is that I think you can use the semi-quantitative readout of an expert or similar molecular test the same way that you use for smear, right? But just knowing that those who have medium and high results are likely to be the most infectious, and those who have low, very low, or trace results are probably more like smear negative patients.
Great, thanks. And I'm going to slip in just one last. I know we're at the hour, but from Penn and Berry at NESCA, will targeted next gen interpretations take data from WHO mutation catalogs, the cryptic consortium, into account, or mostly from internal CDC data? Is there kind of sharing?
Right. We're taking into consideration both. We've been using the catalog, and basically what we interpret is aligns with like all of those high confidence mutations that are in the WHO, kind of aligns what we are seeing in our data. For some of those that are intermediate, we may report some of those as effect unknown currently, because we have not seen them often enough in our, in what we have tested here. But the majority, and especially the high confidence, yes.
Terrific. Well, I know we're a minute past the hour, and I just want to thank you both for joining us. This is just such a great little taster of what's coming down the road, and we're all really anxious, and we thank you both for the work that you're doing here. Thanks for joining, everyone. I'm sorry we couldn't get to all the questions, but it's really good stuff coming down the pipeline. So thank you, and have a good rest of your day. Bye.
Thank you. Bye.